Hello, this is Gina Piscatelli with a lecture for Anatomy and Physiology 2 students at Madison College. And this lecture introduces you to adaptive immunity. Some people call it true immunity versus just the resistance mechanisms that we studied last week. This is the first lecture for week 13 of our semester. And this cartoon shows you one feature of adaptive immunity that differs from resistance. So this cartoon reflects the fact that someone's body was exposed to Escherichia coli, otherwise known as E. coli bacteria. And it must be the second exposure at least, because there is a memory cell that recognizes this E. coli bacteria and can respond to it. And so he says here, I will never forget you. So this memory cell is unique in adaptive immunity, and it's formed due to our first exposure to a pathogen. And then memory cells form, and upon second exposure, the presence of these memory cells make us respond quicker with more defense against infection by this repeated pathogen. So in addition to memory, there are other features of adaptive immunity that differ from resistance. First of all, adaptive immunity is systemic. That means it's occurring in multiple places throughout your body and not restricted to just one site where the infection initially occurred. With resistance, particularly the process of inflammation, we saw that the site of infection is where the most activity takes place, or where a neutrophil encounters a pathogen and engulfs it, or even washing our hands. That's just happening at the skin surface. In adaptive immunity, both the circulatory system and the secondary lymphoid organs, as well as the lymphatic vessels, and some of your tissues will be involved. You'll also see sometimes a fever, right? So that's going to be systemic. Now, when adaptive immunity kicks in after exposure to a pathogen, the response is slow. It's slower, at least, than resistance mechanisms. Mechanisms that involve things like barriers, chemicals, and the encounter of neutrophils and pathogenic organisms, just involving phagocytosis, is pretty quick. But adaptive immunity takes 5 or 6 days to really mount a full-fledged response that can eradicate a pathogen, or at least decrease your symptoms and eradicate the pathogen. Now, after subsequent exposures, though, that's just the primary exposure or the first exposure. But if you're exposed to the same pathogen again, then as I said, you have these memory cells that can make this immune response quicker and stronger. It'll never be faster than resistance mechanisms. It'll go down to like 3 days. But it's enough and strong enough that it protects us against the fiercest pathogens. Another feature is that we did talk about lymphocytes in resistance, but only the natural killer cells. We didn't talk about these other types of lymphocytes, B lymphocytes and T lymphocytes. They're still white blood cells, but they're only involved in adaptive immunity, not in resistance mechanisms. Another significant feature of adaptive immunity is that we say it is specific for a certain pathogen or pathogenic antigen, something harmful to us. The response is specific. What that means is the response will be tailored to the certain pathogen that entered your body. It won't be exactly the same set of cells or chemicals that are activated as a different pathogen might elicit. So, to explain specificity, we'll talk about it in more detail subsequently, but it involves activating just a subset of all the B lymphocytes you have in your body, and subsequently it activates only a subset of all the antibodies that you could produce. You're only going to produce antibodies for that particular antigen once you're exposed. In addition, the T lymphocytes that you have in your body are more numerous than will be activated by one particular pathogen. So, just a subset of T lymphocytes will be activated, and that subset will be particular or specific for that pathogen. So, in summary, the features of adaptive immunity that are very important are in opposition to resistance is that adaptive immunity is systemic. It occurs body-wide. It's slower to mount a full response than resistance mechanisms are. The response is specific, involving just a subset of B lymphocytes and T lymphocytes that are both very specific for an antigen, a specific pathogen, I'll call it. And memory is created so that the next exposure will be quicker. Okay, I've used this term pathogen quite a bit, but the reason that we can activate an immune response, or the reason an immune response even occurs, has to do with molecules called antigens. And these antigens are present on pathogenic microorganisms, or they're free-floating chemicals that can enter our body, like bacterial toxins. So we really define an antigen as any molecule or molecular compound that binds to an antibody, which is shown here as this Y-shaped molecule, or binds to a T cell receptor. And a T cell is a lymphocyte, and lymphocytes have little receptors on their membrane. And so an antigen can bind to that. An antigen can also bind to an antibody, but with specificity. So there are many antibodies and many T cells and T cell receptors in your body, but one particular antigen will bind to only certain types of antibodies or certain types of T cell receptors. The term antigen comes from scientific discovery of the fact that certain chemicals or compounds generate antibody production in your body. And so antigenic means it generates antibodies, but now we know a lot more. It also causes a whole host of actions by T lymphocytes. But that's where antigen comes from. So there are different types of antigens that you could be exposed to that would cause an immune response. Some antigens, as I mentioned previously, are soluble, and that means they're circulating in your body fluids, like in your interstitial fluid. It could be even in your gastrointestinal tract. It could be in your interstitial fluid, in your bloodstream, whatever. And some examples would include toxins released by a bacteria, or maybe a free virus. Viruses are really, really small. I can't really draw a virus very well, but I can try. Viruses are really, really small, but they're floating. They can float in your body fluids, mucus, everything. There are some antigens, though, that we are exposed to that are actually attached to what we would call a true cell or a microorganism that could live on its own. That would be like a bacterial cell. And these antigens are on the cell membrane of a bacterial cell, or it could be your own cell that's infected with a virus, and now your cell has little viral particles on its membrane because it's been infected. And so that could be recognized, and that's called a membrane-bound antigen. In addition, another example is that when our cells become cancerous, they produce abnormal proteins that get inserted in the membrane. And so that would be another example of a membrane-bound antigen that the immune system could respond to because it's abnormal. So the mediators of adaptive immunity are going to be antibodies that bind to these antigens that are dangerous or pathogenic. Antibodies could be attached to B lymphocytes. Some lymphocytes just put the antibodies on their cell membranes, and then the antibodies bind to a pathogen. Or antibodies could be secreted by certain B lymphocytes, but then we call the B lymphocytes plasma cells. So a B lymphocyte that secretes antibodies is called a plasma cell. So B lymphocytes and plasma cells fight with antibodies. And because these antibodies and B lymphocytes are circulating in your body fluids, this arm of adaptive immunity is referred to as humoral immunity because the body fluids were at one time known as humors. Like blood is a humor, cerebral spinal fluid is a humor, etc. So adaptive immunity has two arms: humoral immunity, which involves antibodies, and cell-mediated immunity, which involves T lymphocytes. Now, there are two types of T lymphocytes that we will encounter when we actually talk about T cell-mediated immunity. We'll do that. We'll talk about it in a lot more detail later. But the two types of T lymphocytes are cytotoxic T cells and helper T cells. But both of these T lymphocytes have this thing that I mentioned previously called a T cell receptor that binds to the pathogen or the antigen right there. And once it binds, it's activated, and a host of things are going to happen within the body to fight against further infection with this agent. So cytotoxic T cells are also called CD8 positive cells. CD means clusters of differentiation. It doesn't matter. The point is that there's another little molecule close to the T cell receptor that somebody named CD8 protein. And cytotoxic T cells that have this CD8 protein are going to behave just a little bit differently than helper T cells that have a different protein or cluster of differentiation called a CD4 protein. So helper T cells have a T cell receptor for a pathogen, but in conjunction with a different protein. Your body has cytotoxic T cells and helper T cells, both, that respond to the same antigen that you've previously been exposed to, if an adaptive immune response occurred. You know, like when you get a vaccination, for example, helper T cells and cytotoxic T cells have both been formed in your body, as have been memory cells that make antibodies against it. And we'll talk about that in more detail later. So the way that the adaptive immune response works, and usually these two arms work at the same time, is that you're exposed to a pathogen, an antigen. And here's a viral particle. It's red. It looks like a star, okay? And in the humoral arm of the adaptive immune response, a B lymphocyte has antibodies that are specific for that virus, and so they bind. The virus and the antibody on that B lymphocyte bind. That activates this B cell, and the B cell starts dividing, and it makes clones that are identical to itself. So now you have a whole host of B cells, B lymphocytes, that are able to respond to more viral particles that are identical. In addition, some of these B lymphocytes are going to secrete or release antibodies, not just have antibodies on their surface, but release. So you'll get plasma cells made, and antibodies are smaller and they can float through all the fluids much more easily, and they bind to exactly the same viral particle because they have the same specificity. In addition, a subset of the B lymphocytes that were formed during this clonal expansion, we call it, will make memory cells. And those memory cells won't be activated right now. They're going to go live and take residence up in the lymph nodes and wait for the second exposure or the third exposure. So that's what happens in the humoral arm of the adaptive immune response. Now, the cell-mediated immune response involves T lymphocytes that recognize this virus, not B lymphocytes. So here we have a T cell that has this purple T cell receptor on it. And rather than being able to bind to a free-floating virus, this T cell has to be presented with the virus by one of your body's cells. We call these host cells that have already engulfed a viral particle or been infected by a viral particle, processed it in some way, and then presents that some part of the virus anyway out on its surface. So T lymphocytes have to have this virus presented to them by one of your body cells. We call these antigen presenting cells, and that's what the green cell would be an antigen presenting cell. But from there, things are remarkably similar, at least initially. So this T cell that first has an antigen presented to it, it becomes activated, just like a B lymphocyte would be, and it starts dividing in the same fashion. So you have a clone of identical T cells with the exact same T cell receptor on their membranes. Some of these cells are going to go out into the body and start recognizing other infected cells, maybe kill them, maybe tag them for the further processing, and we'll talk about that later. But there'll be actual killing of your host cells that are infected by these T cells. That's okay, though. Your body can make more of the same cell type through mitosis. But anyway, beyond making just more of themselves, some of these clones will become memory cells and go live in your lymph nodes or whatever, and wait for the second exposure. So a feature of both arms is something we would call clonal expansion, and that's where the cell division process takes place after exposure, and memory formation, which also happens after exposure. Now, I mentioned that during the course of a normal immune response, it actually takes some time. It's a little bit slower. And these are the steps that explain why it takes like week to 10 days to actually see a decrease in the symptoms. And here we've got, first exposure at time point 0. And the first thing that has to happen is the right cells have to recognize that you've been infected. So you have to have B cells respond, and you have to have antigen presenting cells present the antigen to T lymphocytes. Then the clonal expansion, or I guess I should say that lymphocytes are activated, and then you get this clonal expansion of all these cells that can fight the numerous pathogens that have entered your body. Eventually, of course, you're going to have the formation of plasma B cells that produce lots of antibodies that can float throughout all the fluids in your body. And like 10,000 per second antibodies are made by each cell. So like quantities of antibodies. And so by day 7, you can measure in somebody's blood that they have a titer or a certain quantity of antibodies in their blood, as well as B lymphocytes and T lymphocytes that are specific for that antigen. And then the killing processes take place, or the neutralization process takes place, trying to eliminate the antigen. And that happens, somewhere between 1 week and 2 weeks, getting rid of everything. But the symptoms usually start declining at this point. And after the antigen has been eliminated, generally speaking, you feel better at 2 weeks. That time period will be shorter for further exposures. But afterwards, the lymphocytes that were active, they eventually, we call it contract, but what they do is they undergo apoptosis, they die. And macrophages clean up the debris. But upon second exposure, you still have these living memory cells. And so these memory cells do not contract, they do not undergo apoptosis. And so that's the power of the adaptive immune response. So it might seem a little complicated, but I have a couple of review questions, or a few review questions that might help you understand what was most important in this presentation. Let's look at the first question. Which would protect you from an infectious agent, meaning a pathogen, faster? An innate mechanism or an adaptive immune response? And the answer, of course, is an adaptive immune response. Let's look at number 2. Why is adaptive immunity considered specific, but innate mechanisms aren't? Well, to let you think about it, you can pause for a while. But innate mechanisms, resistance, happens the same way no matter which pathogen it's fighting. Adaptive immunity is considered specific because just a subset of cells and chemicals are used for one infectious agent. A subset of what you have available in your arsenal is used for each different pathogen. Let's look at number 3. If an infection first occurs in a cut on your finger, and then it spreads, maybe the bacteria multiply and spread through your lymphatic system or your blood vessels, would innate mechanisms or adaptive immunity fight the infection at distant sites away from the original infection, fight the infection better? Which would help fight the infection better? And the answer is, of course, adaptive immunity, because it's systemic. And then the fourth review question, what are the 2 most important distinctive characteristics of adaptive immunity that are lacking in innate or resistance mechanisms. Maybe this should really be 3, but all the experts say there's 2 most important features that are distinctive for adaptive immunity. And that is, or they are, specificity and memory. Those 2 features, specificity and memory, are distinctive characteristics of adaptive immunity. Resistance doesn't have those. But I would argue that systemic is also maybe another one. But the other 2 are the ones that people classically think of. Okay, I do have some user-friendly videos to post for you to listen to, so you don't have to listen to me talk about this material. And I'm going to post these sites for you. One is the Khan Academy and one is the Crash Course. They both put out really good videos on YouTube for the adaptive immune response. Thank you.